Assalamualaikum warahmatullahi wabarakatuh and good morning to all examiners. My name is Ezatul Syazwani Binti Muhammad Ismail, student bachelor degree of Science with honours of Aquatic Science. Today, I'm going to present my final year project proposal. The title of my final year project is Detection of Redclaw Crayfish, Chirax quadricarinatus, Juan Martins, 1868, using environmental DNA at Felda, Tenang, Besut, Terengganu. My supervisor is Dr. Fathurrahman bin Lananan and my co-supervisor is Dr. Ahmad Syazni bin Kamarudin. Here are the content of my presentation today. First of all, for the introduction, invasive alien species is a non-indigenous or alien species that invades marine and estuarine ecosystem. They also can be defined as the species that have been translocated beyond their natural or historical range, either accidentally or on purpose. Their existence can contribute to the extinction of native plants and animals, damaging the biodiversity and permanently altering ecosystem. Thus, they can lead to significant disturbance of marine and terrestrial ecosystems. Next, we move to the background of study. Chirax quadricarinatus is a species native to northern Australia and southeastern Papua New Guinea river catchment. In Malaysia, they are locally known as freshwater lobster. Based on Awangku et al. 2016, these redclaw crayfish were introduced in Malaysia for aquarium and aquaculture industry. Thus, the growing culture sector of this species will facilitate the spread of this species in Malaysia. Environmental DNA is the DNA that is detached from the organism and is released to the environment. eDNA also is a collection and analysis of an evolving conservation tool that shows promise to improve the detection of freshwater species, especially invasive species. Next is problem statement. According to the previous study at 2016 by Awangku et al., the hatcheries and small-scale culture facilities of the redclaw crayfish were recorded in Terengganu, Perak, Pulau Pinang and Negeri Sembilan. Since Felda Tenang is one of the residents at Terengganu, the spread of the redclaw crayfish are probably due to the escape from the hatcheries and aquaculture industry. Significance of study. Chirax quadricarinatus is one of invasive species that exists in Malaysia. According to Awangku et al., in their study at 2016, they found that the fishermen suffer loss due to the damage caused by the crayfish to their cage and fishing net. While for the eDNA, eDNA is quickly gaining attention as tool for ecological survey. eDNA tests can be performed for multiple species using single sample and it provides early detection of invasive species such as Chirax quadricarinatus. Here are the other significance of my study in terms of authorities, academically, locally, environmentally, and economically. Objectives of my study. First, to compare and analyze environmental DNA water sampling method in Felda Tenang, Besut. Second, to determine the presence of Chirax quadricarinatus in Felda Tenang based on eDNA sample and its results. We move to the next part, which is literature review. For the study of invasive species at 2003, Hussein and Anamalai state that invasive alien species are animal or plants from another part of the world that do not belong in their current habitat and they are intentionally or unintentionally introduced to the local ecosystem. At 2008, Chandra and Gerhard found that this invasive species is eating up resources used by native species, causing damage to local ecosystem and people, or directly threatening native species. For the Chirax quadricarinatus, at 2002, Egerton found that this species can act as disease vector from harmful microbes. At 2003, Katz and Ferrer found that this redclaw crayfish shows the overall trait of competitive invaders of crayfish with large dietary requirements and high reproductive capacity. And at the year 2016, Awangku et al. State that the fact that no reports of environmental damage caused by the crayfish in Malaysia are currently available will probably be attributable to the underestimation of bioinvasion risk issues and the lag period in the observable impacts and species establishment. While for the study of eDNA, ponds were the first natural habitat screened by the researcher who demonstrate accurate identification of invasive American bullfrog. Then, at 2011, the studies of eDNA have shown an improved likelihood of eDNA detection over conventional Asian carp and American bullfrogs. At 2013, Pilier et al. Found that eDNA implementation of methodologies for qPCR, which is quantitative polymerase chain reaction. In 2015, the researchers found that there is large and increasing number of studies have utilized eDNA in a variety of environment. Then for the past 4 years, Denner et al. Found that eDNA is more cost-effective and logically feasible than traditional counterpart. Lastly, for the recent study at 2019, Harper et al. Found that this tool has a huge potential to inform aquatic conservation and management. Next, I will explain to you about the methodology that will be used in this study. Firstly is field survey and eDNA water sample at Felda, Tenang, Besut. Then, DNA extraction will be done. After that, the sample will be going through polymerase chain reaction, quantitative polymerase chain reaction. And finally, the data will be analyzed using chi-square automatic interaction detector. I will explain further about my methodology. For the first methodology, eDNA water sample will be collected at Felda, Tenang, Besut. 3 replicate sample of water will be collected using 100, 300 and 500 ml syringe. For DNA extraction, eDNA will be extracted from the filter using Kajen according to the previous study at 2019. Each filter will be treated, then there are solutions that will be added to the recover solution. The step will be repeated until the mixture is completely processed. DNA solution will be stored at -20 degrees Celsius. The PCR solution consists of primer, kappa biosystem, DNA solution and distilled water. The water sample will be used as positive control and 1 liter of distilled water will be used as negative control to confirm that the sample was uncontaminated. All the PCR will be duplicated and the PCR products will be visualized using 2% agarose gel. Next methodology, qPCR will be used to confirm the presence and abundance of Chirex quadricarnatus DNA in the water samples. The quantification of the DNA copies will be created using DNA cloning. DNA cloning will calculate the number of copies and will be used as a template for the standard curve. Lastly, for the data analysis, chi-square automatic interaction detector decision tree will be used to explore the effect of capture and storage qPCR Chirex quadricarnatus value. This chi-square automatic interaction detector is a non-parametric tree building method that can handle multivariate categorically induced quantitative response. At the end of the study, there are 2 expected results that will be observed. First, the potential Chirex quadricarnatus distribution in Felda Tenang will be successfully located. Second, the presence of Chirex quadricarnatus in Felda Tenang based on eDNA result can be determined. Here are the Gantt chart for the flow of my final year project. And here are the references that have been used for my presentation. I think that's all from me. Thank you and see you next time.